Okay, I think we need to get started again. Everybody, please sit down. We're moving on now to talk about the challenges of immunotherapy and how to select patients, which was something that came up yesterday. It's a hot point of debate, and we're going to start with 3 different viewpoints on virological, immunological, and clinical biomarkers for selecting patients, and then invite a panel up for a discussion about this. So I'm very pleased to invite Barbara Testoni to start off. She's a PI in the viral hepatitis team at CRCL INSERM in Lyon. Barbara, you're going to give us the rationale for virological parameters in selecting patients for immunotherapies. Thank you.
Yeah, thank you so much. Thank you so much, Mala and Antonio, for this invitation. And also for pushing me a bit outside of my comfort zone this morning. So trying to look at these biomarkers from a different point of view. So let's see what we can get from it. So first of all, what we can measure and where we can measure. So we can measure viral parameters in the liver with different levels of sensitivity, specificity, and technical challenging. We can measure cccDNA, replicative intermediates. We can measure, to some extent, integration. We can measure all the RNAs, and of course, we can measure proteins. In the serum, we can also measure the DNA, the antigens, and also, as you know now, also the RNA. So for the sake of time, I will stick to the serum compartment in this flash talk. But I would like just to point out the HBV genotype, which we cannot usually talk about it as a marker, but indeed it's a viral feature that has impact on the viral replication, pathogenesis, and so most reliably also on immune responses of the host. So beginning with a very simple concept, we all know that viral antigens play a pivotal role in modulating host immune responses. And so I will really look at some concept on 3 antigens biomarkers, so S, correlated antigen, and then circulating RNA. So HBS antigen, so begin from the simple, not so much, you will see. So loss of HBS antigen is really the hallmark of functional cure, together with the sustained loss of viral load. And this functional cure is associated with the residual virus in the liver, meaning that a persistent immune control is needed to reach this functional cure. So if we can get something, for example, if take as an example the STOP NUC studies, and we compare the patients who reached HBS loss or didn't reach it, and just not taking into consideration all the heterogeneity of cohorts, criteria for retreatment, so on, it's clear as a day that the restoration of immune responses, which is supposed to happen when patients lose the HBS, is associated to the lowest HBS levels possible at the moment of stopping the treatment. And then there's a debate about this threshold for the selection of patients. And as you have heard from Ulla this morning, the lowering of S antigen is really implicated in an optimal response to therapeutic vaccination, at least in a proof of concept in a preclinical model. So many questions remain, because this seems quite simple. Okay, low HBS, good immune response. It's a bit more complicated than that. For example, we don't know if there's a difference if HBS low levels are acquired spontaneously or naturally, or if they are therapeutically induced. And should we need to lower HBS in the serum? Should be sufficient, or should we act into the liver? And should we measure the S antigen components, L, M, and S? Moreover, to make the picture a bit more complex, Nina Lebert showed that HBS-specific T cells reduction was more associated to the duration of of exposure to HBS than to the overall quantity of HBS in the serum. So putting the length of duration of infection of the patients and thus the age in the picture. Moreover, a very recent paper showed that age and correlated antigen rather than HBS are associated with HBV specific T cell responses. And so another actor comes. Correlated antigen, what is it? So we know it's a composite marker without entering into details. It has been really thoroughly shown that it is associated with intrahepatic cccDNA transcriptional activity, may predict cancer incidence, may discriminate active from inactive HB antigen negative patients. And indeed, I believe it is able to identify a subset of patients, in particular within the HB antigen negative patients that cannot be identified only by HBS and levels of HBV DNA. The characteristics of these patients are to be explored because it's not very clear from what we get now. So probably immune markers could help us in understanding what correlated antigen could bring in. And indeed, bring back to the paper that I cited before, the author showed that patients with low correlated antigen showed a higher HBV-specific CD4 T cell responses, and especially in core-specific and pol-specific. And moreover, these CD4 and CD8 T cell isolated from patients in vitro showed an increased responsiveness to PD-L1 blockade. So in the case that the patient had very, very low level of HBS and correlated antigen, but also when HBS levels were a bit much higher, but with very, very low correlated antigen. So then we have serum HBV RNA. So I don't have much to say on that, unfortunately. So we know that it well correlates with intrahepatic cccDNA activity in preclinical models and in patients. It's a very, very promising marker for the intrahepatic activity of the virus. But we have a big problem. There's the heterogeneity of the assay, and most of the assay are in-house assay, and they do not recognize the same RNA population. This could have a big impact on the readout because you can, switching of assay, you can have positive or negative results and a wide range of levels of quantification, of course. So even if we have a couple of things coming out about the correlation of serum antigen and immune response, beyond the biological and clinical value of that, the question that comes to my mind is, okay, but would I had the same result if another assay was used? So coming back to the stop nuke studies, I think that another thing that we can learn from this type of approach is that both correlated antigen and serum HBV RNA associated to S could help in better stratifying the patients for the probability of achieving or not a functional cure. So in which context and in which type of therapeutic intervention has to be discussed. So just final sentence to say that probably we are not ready to move away completely from the liver. So I think we will discuss it later on. We have a lot much to learn. Even if from the virological point of view, the biomarkers that I presented you are beginning to be well characterized. They are not perfect, but they are very promising. But we really need to couple these new biomarkers with the immune markers to really understand how they work together and their predicted value. Thank you.
Thank you very much. We are not going to run now questions, but we have immediately the second speaker, which is Adam Göring. Adam Göring has been working, let's say, for many years in hepatitis B, first at the University College of London and Singapore together, and now is a PI at Toronto. Adam, you have to talk about the case for immunological parameters.
Yeah. So great. So to get started, I've listed all of our immunological parameters to measure here, and provided a reference to the ICE-HBV serum biomarkers paper, which is actually a fantastic comprehensive review of what's available in the roadmap. So anyways, no, I am going to present something, but it's pretty limited, to be honest. Antonio gave this, I could hear him laughing in the email when I got the invitation to speak. But we now have really a wealth of data on the mechanisms that regulate immune responses in chronic hepatitis B, and the challenge is really how do we turn those into a biomarker to select patients for immunotherapy, and do we need to select patients for immunotherapy? And I think in some cases, on the last slide, I'm going to make a case where there are situations where that's going to be necessary. So I would say, of all the stuff that we've measured in terms of immunological responses, and I didn't update this maybe from Mala's data presented on the B cells and the stop nuke, but T cells have really been what has been associated with control, and I put Antonio's paper here on purpose. And these are looking at patients stopping nucleoside analogs, and the idea is, can we use an existing T cell response to predict control? And I would say, it's possible. These patients in this study had more core and polymerase-specific T cells. They didn't have flares after stopping antiviral therapy. And so these are seen here. Unfortunately, the cutoff line has been eliminated here, but basically anybody above this line were the ones that were the only ones sort of above the background. So you can see that the patients that had a larger response, the T cell response, had less inflammation after they stopped nukes, but this was also a significant proportion of patients underneath the cutoff line that they identified here. And one of the issues around this was that in this study they're using in vitro expansion, which has been quite useful in characterizing the response, but it also requires a significant amount of labor. You got to take the T cells, put them in culture, load them with peptide, grow them out, acquire them, and then Massimo can give us a thesis on user-dependent gating and analysis in flow cytometry, because it is dependent on where they're being gated. And so this leads to a bit of variability that's not really usable in a clinical study, or particularly for patient stratification. And so one of the things that we did in my lab is trying to at least improve on an existing assay, which is the ELISpot or fluorospot assay, to try and get an ex vivo response. And so in this case, this is looking at patients across the different phases of hepatitis B, including functional cure, and looking at the interferon gamma response. And you can clearly see that in a good number of patients, not all, but in most, we can actually detect an ex vivo specific HBV specific T cell response, seen here in the filled circles with the HBV peptides. And I think the most important aspect of this is you can look at it more than one cytokine directly ex vivo, and I think this is going to be actually quite important, the ability to measure IL-2 responsiveness, because there's more data coming out. And I was working, collaborate with Georg Lauer, showed recently that CD4 cells were important, so I think a lot of this IL-2 that's coming up in these patients with good viral control may be coming from the CD4 responses. And here, when you think about an ex vivo assay, I won't say it's easy, right? It's still very challenging to do because there's still a lot of components that go in, but everything goes in the plate. You wait 24 hours, you acquire spots, and you count spots. And there's still some parameter setting, but it's more straightforward than flow cytometry gating. And so now we think about this and how we should use a potential pre-existing T cell response to stratify the patients. And I'm only going to cover a couple of topics because I think only a couple are really relevant for this, even at this point. But in terms of therapeutic vaccines, I really don't think there is a patient selection criteria here because of the specificity of these vaccines. We don't know who's going to respond, so there's no reason that we should start with a patient with an existing response. And there's little chance of an off-target toxicity. And you think about the timing of how to deploy these immunotherapies within these treatment regimens that are coming down the line, siRNAs, ASOs, CAMs, any of the new the new drugs. I mean, in monotherapy, there's no consideration because you just start these patients. And in combination, what it looks like is that with siRNA and ASO, these reach nadir at about 6 months after therapy. So they reach their sort of baseline of HBs antigen. And I think really starting the vaccination 3 months or so before the end of the siRNA, so you try and time the peak in the T and B cell frequency at therapy termination with the siRNAs, is going to be important. And I think data is coming up now, particularly for checkpoint inhibitors like PD-1, that probably at the final vaccine boost, and this is coming out of some of the Vaxitech data and also some mouse data, instead of doing PD-1 repeatedly, probably at the boost is likely to be the most effective. And so moving to this idea, these patients with checkpoint inhibitors now, and this becomes more relevant because here patient selection should factor into who's going to get the therapy because it's relevant because of immune-related adverse events. And the idea would be to exclude patients that are susceptible to autoimmunity, and right now they do autoantibody screens, but really try to include or enrich for patients that are going to respond to PD-1 blockade. And in terms of therapy and monotherapy, I mean, again, there's no time consideration here. I think probably patients without elevated ALT would be the only thing. And combination with ASOs and siRNAs, again, here I think it's probably more likely at the end of dosing when you start to get this slow rebound in virus production to again, just capture changes in the virus-host interaction. So in the context of patient selection for PD-1, PD-L1 therapy, I think that the ex vivo T-cell response, again, it's not easy, but it might have some indication. And we've built on the ex vivo assay to then not just do HBV in specific responses, but this is isotype and anti-PD-1. And this is looking at interferon gamma responses across the different cohorts of hepatitis B to see which patients actually respond best to PD-1 blockade. And to be honest, interferon gamma was a fairly poor indicator of PD-1 responsiveness ex vivo. And what we found is that IL-2 was a much better indicator of PD-1 restoration directly ex vivo. So I think just measuring interferon gamma is also not the right way to go. And I would always prefer to do both cytokines or at least IL-2. And then, trying to relate this back to patient characteristics, it's maybe not so exciting, but the e antigen negative patients have significantly more restoration when you block PD-1 in both interferon gamma and IL-2, and even at this kind of the baseline responses in IL-2 are much higher in these patients, so they might be much more likely to respond. But again, coming back, these are difficult assays to do, even in immunology labs, to detect these responses. So what we really need to do is get down to an assay that doesn't rely on HBV-specific immunity because it is so hard to measure. And so I'm working with a colleague at the Princess Margaret Cancer Hospital in their oncology unit, and they're doing a lot of immune monitoring where they're looking at, this is, again, a response in the PBMCs to type 1 interferon. And so in this case, it's much more of an adaptable clinical assay where they have the total PBMCs, they expose them to interferon beta, and they have a CyTOF panel in which they're actually measuring an ISG profile. They did it in, 30 different populations, but in the end, what they found was, that came bad. In the CD4 effector T cells, so these are CD4 effector T cells, they saw pretty distinct profiles of response. There were CD4 effector T cells with a high interferon response capacity, which is this y-axis here, and then a low interferon response capacity. And they took this interferon response capacity and compared that to overall survival in checkpoint blockade patients. And what they found is really a very distinct survival curve. So these patients that had a low interferon responsiveness at baseline had much better overall survival. And so, there's a big leap from saying patients with overall survival relates to patients that have a better HBV-specific T-cell response to PD-1, but I think it indicates that there's the potential to get a non-virus-specific biomarker for hepatitis B. And I think that's all I'm going to put up for today, and I'm happy to take discussions. I didn't put anything up about the liver, but hopefully we can have some discussions on that in the debate section or the discussion. Thank you.
Thank you very much.
Thank you very much. That's great. And now we've moved on to our final case for clinical parameters. Uppy Gill, who's from Barts and the London, consultant hepatologist and immunologist. Thanks for your input.
Thank you to ICE. Thank you, Marla Antonio, for the invitation. Switch my slides.
We need the slides. Okay.
Perfect. Thank you. So thank you to Barbara and Adam for those lovely talks. I'm been asked to talk about clinical parameters, and I'm probably going to merge a little bit of what both Barbara and Adam have already mentioned too. So we've heard a lot about what we've already got in chronic hepatitis B and what we are trying to achieve. So we have already met some needs. We've got good antivirals that suppress HBV DNA. They may regress fibrosis and potentially halt cirrhosis and possibly even reduce the risk of HCC, but we don't know if this is true. What we can't do, as we know, is there's no direct necessary effect on elimination of cccDNA, the rates of surface antigen loss, even if we think that is the holy grail, is not really achievable with the current antivirals that we have at a good rate. And long-term therapy is still required with the issues of potentially safety and cost, especially in low-income areas. And what we are trying to essentially do is boost immunity correctly, not that we then get overwhelming flares, but also to try and reduce the intrahepatic HBV burden. So clinically, when we see a patient, when at least on a new diagnosis, we do an array of tests, and then we continue to follow them up at least twice a year, if not more, depending on the sort of disease phase that they fall into. So we would do some serological tests using e-antigen, checking if they have an e-antibody, and this is once they're known to be surface antigen positive. Check the HBV DNA, exclude any other infections, and then it's really the biochemistry that I guess is the clinical parameter that we have. So we are then just measuring ALT and HBV DNA 6 monthly, and with that, we want to know that they've not got any evidence of decompensation, so that their synthetic function is okay. And that really you want to do when the patients have got more advanced disease. Of course, measure an alpha-fetoprotein and get some imaging to make sure that they've not got any risk of or developed a space-occupying lesion or a cancer. And in terms of measurement of fibrosis, yes, we can do a liver biopsy, but now a lot of centers go for transient elastography, so just measuring a FibroScan result. So in terms of clinical parameters, yes, I would say that the minimum we would have is measure transaminases, a HBV DNA, a surface antigen level if we have one, because that is useful, and then also some image of the liver. And then in treatment-naive patients, yes, we want to continue to know if they are going to spontaneously seroconvert, so you measure their e-antigen along with everything else I've said. And then on treatment, there are various other things to measure depending on the treatment that we are using, and they are listed here. And in terms of how we currently treat and how we select patients for the future, well, it's all really been based on disease phase, which I've both put the old terminology and the new terminology here, and I'm not going to go really into that because I'm sure we all know about this. But really, treatment at the moment is in those phases where there's an elevation of transaminases, or at least or an elevation of the HBV DNA. In the phases where there's sort of quiescence of ALT and HBV DNA, or these immune control phases, treatment is still not really given. Now, that may not be correct, and we could be just treating anybody here, all patients, in fact. You know, if you're surface antigen positive, we could give everyone a drug and then think how we select them then with immune therapies, rather than just using an immune therapy when we've got no nucleoside analog or not lowered the viral load initially. But what about some other things? So then, is using the ALT, the HBV DNA and surface antigen level enough? Will that be enough in selecting patients? What else do we need to do? Do we need to consider these parameters with any other parameters that Barbara and Adam have mentioned? And what about co-etiologies for patients? So firstly, the patient age. I'll come on to talk about that in a moment. Disease phase, how do we then put them in a disease phase? We've got these, let's go back. Let's just go back. We've got these disease phases. Is that important, how we choose patients going forward? Do the patients even want to be treated? And then the other thing is cost of drug and feasibility. I did just want to go back to co-etiology, because a lot of patients where we're looking at ALT now, the ALT elevations may not be due to hepatitis B. They have very low levels of HBV DNA and they have a raised ALT. Is that because they've got some amount of fat in their liver? So in that case, at least for some of the trials that are being selected at the moment, there'll be a plethora of patients that are completely excluded because they have mild amounts of fatty liver, and therefore they wouldn't be candidates for clinical trials, at least at the moment. But actually it's not their hepatitis B that's causing that inflammation, or at least we don't think it is. It's another etiology. So how are we going to choose and select those going forward for immunotherapies? Now, we did some work basically looking at some viral markers, and I'm wanting to show you this just in terms of correlation of clinical parameters with viral markers, which Barbara has already mentioned. And here we can see that we looked at intrahepatic HBV DNA, and this is all in a cohort of the antigen-negative patients, pregenomic RNA and cccDNA, and we're able to see that these correlate quite nicely with HBV DNA. So potentially we could just use HBV DNA as a surrogate here, and that may be enough. But actually, as Barbara had said, the other parameters do tell you something else about what's going on potentially in the liver. So what we need more so is an accumulation of these, but probably the single best marker going forward to make things less complicated. This study's also been mentioned. This is work that we did, myself, Nina, Michelle did in Antonio's lab. So we had showed here that it wasn't actually surface antigen levels, but the duration of infection that had an impact on the immune response in terms of the surface antigen specific T cells as they underwent attrition over age. So as we've been talking about, looking at patient age and treating patients earlier may be beneficial. Now, the other thing about clinical markers is when we're doing experimental work, we're always trying to correlate it with some immune or virological parameters. And we've seen that, for example, these are just a couple of examples that trail-positive NK cells may correlate with ALT flares. So that gives us an idea of something going on in the liver. HBV-specific T cells may correlate with some surface antigen loss, but again, that is also dependent on what peptide you're looking at, how long the patient's been on treatment, and other factors involved with that. So this is all work that's essentially been done in the blood, but is it still important to look in the liver? Because that's really where the action is happening, but we will need some peripheral correlates for that. So just going back to a bit of work that we did with Laura Pallett in Marla's lab, looking at these fine needle aspirates and the importance of tissue residency, which has been already mentioned, that we were able to show that we can detect tissue resident T cells from a fine needle aspirate, which were essentially equivalent to that seen in a liver biopsy, and that's both for T cells and NK cells, which were different from the blood. Now, work that we've been doing with Barbara Testoni and Fabian Zulin, we've been also able to essentially show the same, that we're able to detect cccDNA and HBV RNA, which is equivalent with a biopsy and a fine needle aspirate. Now, I'm showing these data not that they're clinical parameters, but to show that these are the things that we need to look at and what clinical links can we make with what we have? I mean, with this, we didn't really see any, but moving forward, we need more of this data to see what are the clinical parameters that can link with our immune and viral markers. So in summary, we certainly need some further studies on natural history, I think. We do need to know who should be treated. Should we just widen the treatment candidacy pool, do what we do in HIV and treat everybody? Or should we really just focus on the ALT? But I've just said also the limitations of that. We've heard that we will probably require combination therapies, but we're not going to know which ones are best until we start using them. And we need to evaluate the immune and viral parameters that we've heard about with the clinical parameters. It will be important to know which agents are efficacious and how to select the right patients. Now we've had an idea of both viral, immune and clinical parameters, but we've got to be probably quite succinct in how we use these and not have 20 different markers going forward. So, like I said, we would need a combination of these and a reliable peripheral marker to show the surrogates of what's happening in the intrahepatic responses is probably what's going to be required moving forward. So I think there's lots of work needed in that. And with that, I'd just like to acknowledge people that have helped me in some of the data that I've presented, and thank you all for listening.
Thank you. We would like to invite all the speakers, and in addition to the speakers, we would like to invite also Simon Fletcher and Andrew Valiant to have, I would say, 20 minutes discussion, 10 minutes more than what is. Just short introduction of Simon Fletcher and Andrew. Simon Fletcher is the Executive Director at Gilead Science. Andrew instead is the CEO of Replicor. And maybe we would like immediately to start with the, let's say, representative of the companies, and with just a simple question. It is, how do you select your patients at the moment for clinical trials? And whether some of the parameters that we have discussed today could have been of use for you.
Okay, I think, the heterogeneity both on the viral and the immunological side have sort of been well described this morning. And I think there's a lot of retrospective analysis of these things, but unfortunately right at the moment we don't really have a great sense of what patient populations will respond the best. And so by default we go broad. E negative, E positive, on nuke, not on nuke. Most of the studies start on nuke treated, obviously for safety, and they expand slowly outwards. But then, of course, the biggest gain will be made in those patients that aren't currently treated, and so then there's additional studies there. So, it's a very nice idea to do all of this. There's certainly things that are done retrospectively, but really not really integrated completely at this stage of kind of clinical development where the response rate is still relatively low.
Can I just follow up? But when you've done immunotherapy trials, such as the PD-1 vaccine, have you been more selective in that situation?
Well, safety was the key thing for the early PD-1. So again, it was an E-negative, nuke-suppressed patient population, and that's what's been done mostly. I think as we go forward, yes, I think that those additional considerations will come in. But I think particularly for the first PD-1 trial, I think the concern was, sort of some of the autoimmune, some of the other sort of side effects that have kind of been raised. So safety first, and then you expand later.
I would say in our early trials, we really just focused on excluding patients with surface antigen less than 1,000. And the reason for this was the data that existed from interferon, showing that with lower levels of surface antigen, interferon response was better. At that time, we weren't sure about our oligonucleotide being immunostimulatory or not, so we wanted to exclude this. Where I think these particular analyses are going to become very useful is understanding why some patients get functional cure and why some patients get partial cure, even with S loss, even with interferon. I think that's where this is going to become very important in figuring that out.
Thank you. Okay, I was asking if there were questions from the audience, but there is a question.
A question for Simon and Andrew. Does the FDA give you a hard time picking patients based on the relative levels of S antigen, because after all, they would like to have a representative population.
Well, I mean, I think we don't really pick patients based off S antigen to begin with, certainly. I know there's been some recent data looking at the lower S patients, and we are seeing better responses with certain things like PD-1. But I think it's important that we capture the representative, sort of spread across different patient disease phases.
We've had no issues. I know that when we get into a phase 3, the FDA will want us to have everybody in there, even the low S. But they're very happy with us excluding the low S for phase 2 trials.
Yeah, so I really agree that at this point, I think we should not pick and choose a lot, no matter what the different assays maybe suggest. I think the key is just for the trials going on that you sample well so that we can figure out later what happened. And I think that has been the biggest challenge. The immunology, we can do a lot more now, but with 20 or 40 million cells total, a couple of time points, we will not learn more than a few dots more or something. And so the question is how to do it, and I think that's going to be really something to discuss. Can one cannot do leukapheresis and liver biopsies in a whole trial, but can one carve out small sub-studies with really intensive samples where we have longitudinal data? Then that really teaches us a lot and in depth from few. And I think that's going to be really critical to start in as many trials as possible, because lots of things are going on now.
Yeah. Can I just comment on what you said? Which, in a way, I agree, because, again, we are doing retrospective studies. But again, the problem of retrospective studies for me is that if we are going to test the immunological parameter on retrospective studies with PBMCs that have been, let's say, frozen, thawed, we can lose a lot. This is my idea. And in a way, this is also why we are here, because I am convinced that somehow we need to get better assay, immunological assay, easy to do, something that, for example, Adam has presented before.
Yeah, I agree with that completely, because I get PBMCs from studies from all different sites across the globe, and the number on the vial and the number you get out and the number that are viable from each one, it varies quite significantly. So it makes it hard sometimes to draw meaningful conclusions. But coming back to Georg's point about sampling, I mean, this is one of the main focuses in the immune biomarker discovery paper that we wrote with HBV Forum, is to really try and lay out a plan where, there are certain groups where you don't need to sample them every week or every 2 weeks, and getting these larger draws or doing these sub-studies with FNAs particularly are incredibly valuable, right? And that's what we've seen in our own personal experience too. And I know you have some experience from hepatitis C, getting an enormous amount of data out of just a handful of patients that consented to leukapheresis. So I think this is always something to keep in the discussion and keep in the minds as sponsors think about designing their studies.
Right. I think you just need to anchor, at a couple of centers that can do the sampling well, where you get these, so that you then have high quality stuff to do the explorative stuff, and then maybe you can come up with something simpler. Not sure yet, but.
So, yeah, I mean, I completely agree. And I think maybe to Antonio's point, I don't think retrospective doesn't mean, do it in a year's time. It's more like don't use that preemptively to enroll patients, I think. But certainly, yeah, the only real way to do this is to do it in sub-studies and some focus sites, both for the PBMCs, but also, I think, and this is what, a number of companies are doing now, doing sort of sub-studies to look at liver biopsies, which I think obviously is highly important, really, as we move forward.
I mean, the danger of not doing any selection, though, is that surely you're worried that the agents will fall by the wayside, because, if you don't have an early success, you may not get to then spread it out to a wide population.
Yeah. I mean, yeah, the goal really then is to sufficiently power your study so that you've got enough patients in each of the different groups that you can actually see that small study, that small signal, perhaps.
Yeah,
Maybe 1 second, Barbara, then Andrew.
Yeah, just one comment. I completely agree that we need proof of concept studies within tripartite analysis, and really we have to sit around and discuss what are the crucial markers that we have to analyze in order to be the most efficient as possible with the material that we can get. Because, for example, in the IPQB program that we are running, there's a prospective study with an immune therapeutic intervention, and when we put down the table of how much blood, how much serum, how much FNAs we were going to do, because we want to look at everything, or as much as possible, that's huge. We are dealing with patients, and so not everything is possible. So we really need expertise to guide us to choose the best efficient way to look at these markers and combine them.
I would just, I think, echo Mala's comment, which is to triage early clinical studies with immunotherapies. I think Immunocore's approach is correct. Restrict the surface antigen less than 1,000. Let's see if we can figure out in that environment what the parameters are, and then sort of optimize it so that you can open it up to other patients.
So, George, do you want to? No. Okay.
I would like to make one more comment on the patient selection, because I think for the larger phase 2 studies, the selection may be less critical because you want to see some diversity. But in these focused sub-studies where you want to dive deep, you need to have well-controlled patients, because you can only recruit, like Barbara was saying, you can only recruit a few patients that are willing to undergo an FNA and blood draws and all this. So you need to have the best defined patients you can to get the best data you can out of it to really resolve as much as possible.
I mean, also with immunotherapies, we're always going to be worrying about safety, so we do need sub-selection just for safety issues, no?
But no, that's what I want to say. I mean, there are some criteria we know are important, which is safety. But I think we can also fall in a trap where we pre-select. Let's say we look at some kind of in vitro PD-1 response or something, and we might select wrong. And then the drug is equally dead because we don't see anything, because we just picked based on something that's maybe not that solid. And I think that's just, I mean, it's really complicated. And so I think a lot of discussions will be necessary to really optimize this all.
Carla?
Yeah, no, thank you. Interesting discussion. I was wondering, what does the panel think about the role of the gut microbiome?
That's a clinical factor.
Let's say that is a clinical fact.
I mean, yes, I think it's important, but again, how we can start using it in terms of going forward for patient selection is going to be quite difficult. Do we then have to control for things like diet, or are we talking about, are we going to use it in terms of measurement of, in, let's say, a sub-study? I mean, that's definitely, probably possible to do. But then I know that Adam was saying that we need to, go deep in doing the sub-studies, but I guess there's probably a point at where we have to stop in terms of, then are we just going to get just more heterogeneity of what we're looking at, and then not really move further into developing a single or a few markers that are going to be useful? I'm sure the gut microbiome will probably have an impact, potentially for immune therapies and for safety, but I think we would probably have to limit what we can do.
Yeah, but just in terms of if we see unexpected or heterogeneous responses, like having an assessment of the microbiome might help answer some of these questions.
That's also true.
Yeah. Adam and then Simon.
Yeah, I mean, I think this is, we can learn a lot from oncology here. For one, we've tried to do some of these. It's hard to get poop from patients. It's not an easy thing to ask, actually. It's an uncomfortable situation, even for swabs. But, oncology is teaching us a huge amount because they're doing it in all of the checkpoint inhibitor therapy studies now. They're always measuring the microbiome. And when you think about these innate immunomodulators, it may not be pre-selecting, but you might condition patients for response to TLR agonists by giving them a probiotic when they start the study or something like that. So maybe not for patient selection, but you could modify response to drug by giving some kind of microbial FMT or something of that sort. But there's a whole other realm of complexity in getting those donors and selecting them, because it's incredibly complicated, because I won't go into the details.
Yeah, I mean, I'd say we have done some microbiome work, and it's incredibly complex. And really what it means, I think we can gather a huge amount of information, and we really struggle to actually work out what actually trends and what actually is out of there. I mean, maybe I'd flip it a little bit, and perhaps, I mean, again, learning from the onc field, it would be useful probably to understand whether people have been on antibiotics and things like that prior to that, because that seems to be what the reduced diversity of the microbial flora seems to be, affecting response to checkpoint at least. And so, there may well be some of that correlation with HBV as well.
Yeah, there's been some studies, including some by the panel members, that in patients with normal ALTs, there is inflammation going on in the liver in, for example, immune-tolerant patients. How useful a marker would it be to have something more specific or sensitive than ALT levels at the moment?
I mean, definitely something better would be better, of course. And I get that, you've sampled somebody, you've got that one measurement of ALT, but that's not really a good marker of what's been happening, let's say, in the weeks before or the months before, which is why you may see inflammation if you've done an immunological assay or even a measurement clinically. I think that the improvement then comes in various sort of non-invasive scores that have occurred. But again, they also become quite heterogeneous. And then what do you use? Which is the best one? Some have a better sensitivity than others. Some are more specific. So at the moment, I don't think anything has been, so much better than just the measurement of transaminases. But then we also have the fact of what level of transaminases do we use? Do we vary them with males and females using the priority criteria, or are we just using a level of around 35, 40 for everyone, which may not be suitable for a young female, and that may indicate that there is ongoing inflammation and going back to, which may be relevant to the gut microbiome too.
I think we run into a randomization limit there with the number of parameters you use to randomize. But I think it's something that's important to look at retrospectively, for sure.
Luca?
I remember back in the old days, there were people, I'm thinking about discriminating between surface antigen coming from cccDNA versus the integrants, and there were people studying compositions of sugars and lipids. But that was way before the omics era, and so I'm wondering whether in industry or academia, is there anybody looking at the lipids and sugars in subviral particles, trying to determine whether there are differences between those coming from cccDNA and integrants?
Wow. So if I got your point, so looking at lipids and so on, associated to subviral particles, to discriminate between as coming from integrants?
An old way of thinking saying that the subviral particles coming from cccDNA and those coming from integrants might have differences in the composition of sugars and lipids.
Okay.
And so, but nothing really came out. But that was before lipidomics, glycolipidomics, metabolomics, and all of that. So my question is anybody doing those studies? Because at least that would be, a decent information to have before starting a trial.
That's a very nice suggestion, indeed. Not in my knowledge. So we are looking at the composition and mass spectrometry of the different composition components that we can find in serum of patients. And thank you. We will look at it.
Simon?
Okay, I can ask me.
1 second, sorry. There are some.
Luca, that's a really good question. I know there's some papers that talk about like O-link glycosylation declines more rapidly during nuke therapy. I think there's a way to do this, and you've got me thinking about it now, which is to take really well D-dominant patients, pull out those subviral particles, and compare them to patients that have chronic HBV infection. And maybe there's something going on there. It's hard to know, though, because I think they're both coming out by that HDL pathway in both cell types, but maybe there's something specific going on.
Yeah, we're not actually doing lipidomics per se, although we have done some related to that, but trying to understand, at least on the circulating RNA, whether we can actually understand whether it's coming from integrants or non by reading through to the cryptic or the authentic and looking at the 3' end. So a little bit of a different approach, but maybe in the same direction.
So I think...
sorry, Mark Garney was next.
Okay, thanks. There's been a push, and I think largely coming from the immunology field, to expand the treatment indications, particularly for patients in the immune tolerant or E-positive chronic infection phase, depending on what terminology you want to use. But the results of trials that have included such patients really hasn't borne out a benefit of treating such patients. So my question to the panel is, do you think those patients do warrant a therapy that should be broadened to the sort of global population, or should they only be studied in the context of clinical trials?
I mean, the question is whether, treat immunotolerant patients. And you also said, okay, at the moment we have not seen any great benefit. I will put it this way. For an immunological point of view, these patients that are called immunotolerant are not immunotolerant, and they might have a slightly better response than, let's say, people that have 60 years old. So at this point, I would say this could be the patients where we want really to go specific with immunotherapeutic approach. And I don't think this has been done, except the fact that in reality, the interferon, which is an immunomodulator, is still the one that is working better in young people.
But not young immune-tolerant patients, right?
I don't know.
It works better in young immune-active patients, but the trials that have looked at combination of nukes with PEG interferon therapy, for example, haven't led to a better response rate. So this is the sort of disconnect. Maybe we don't have the right drugs at the moment. So I wonder if perhaps...
different strategies.
I think the issue there, Mark, is not expecting a better antiviral response on therapy, but the recognition that integration begins very early in the infection and is progressive. And I think that was one of the issues with the update we got from the Polaris group at the Mexico meeting, which was we're doing a great job of normalizing ALT and suppressing viral replication, but deaths are rising, probably from HCC. So I think this is one of the things that needs to be considered when we're driving treatment, expanding exposure to treatment in those patients, is trying to slow down the rate of integration.
No, I do get that, Andrew, but my understanding is integration is a very early event, and it probably happens within hours to days after initial infection. So unless you can actually identify when a patient is truly infected, are you really changing the patient's course by treating, after, say, 10 or 11 years after, 20 years, 2 decades after infection?
I think if you shut down the production of HBV DNA in the blood, you're also lowering the chance of rcDNA forming integration. So this is a question about the integration burden over time, lowering that slope. If you wait, let's say we wait until patients we know they're chronic, and we say, okay, let's start treating them. At least then we're getting early enough in the therapy, not waiting until they've had 20 years of integration before we start treating them.
Yeah, Mark,
we have a question. We didn't forget.
I know. I want to just follow up on Mark's thing with one last point, because, yeah, I mean, like Patrick and Antonio, we've seen T-cell responses in the antigen-positive infection patients, but there is something different about them. And I don't know exactly. I think none of us are particularly clear what it is. And it's the same with the antivirals. They the higher levels of starting DNA and S antigen just may have the same effect, but it's hard to get to that baseline. I mean, I would say when you think about these different patient categories, though, and particularly the E antigen positive patients, I think Mala and Antonio and even Frank described it in the 1970s, right? So inflammation, ALT, is probably the worst thing you can do for immunotherapy, to be honest, because it does reduce T-cell or immune responsiveness. So I think this should be sort of when we think about therapeutic vaccines and going in, I would at least be a proponent of making sure that the ALT is not elevated in these patients. I think that, at least when we think about the category, should be key consideration.
Thank you, Antonio. So first, I thought I was telling Luca I could say a few words about this lipid composition, right? And then I have some comments about the viral markers. So with the collaborator we have in the department, we're doing some structural studies on the surface antigen, right? So we just published a study last year. So first of all, those spheres that what we have, they actually don't have any lipid binaries as people always draw in textbooks. Okay? Those are in fact just lipoprotein complexes. Okay? So they all have lipids, but the lipids are very different from lipid binaries. There is no lipid binary in those spheres, right? But whether the surface antigen released because of integration versus cccDNA, I think it's probably unlikely that the lipid composition will be different if they are assembled similarly. Okay? But I remember some studies suggesting that the ratios of L protein to the small protein might be different, whether it's derived from the cccDNA or integrated DNA. Okay, so I think that may be worthwhile to look into. But I have some general comments about viral markers. Okay, like Barbara gave a very good summary, but I think we need to think a bit more sort of in advance, in parallel with current development, right? For example, the RNA markers, it reflects RNA packaging mostly. So if like a capsid inhibitor that's known to block RNA packaging, serum RNA is not a marker for cccDNA anymore. I can sense even among the prominent physicians, there's some confusion about that. Yesterday, we heard about that.
Absolutely. I completely agree with you. We really have to be careful about a marker of target engagement and a marker of response. Absolutely.
Also with chlorinated antigen, for example, right? If you do siRNA, you're going to reduce or hopefully eliminate all antigens. So again, in that case, chlorinated antigen is not a marker for cccDNA either. Okay? So I think we are sort of running out of virological marker either. Let aside the problem with immunological markers. I think that's something we really have to think about, right? So I mentioned some problems with chlorinated antigen, but overall, there's also the sensitivity issue. Okay, so I hear people talking about surface antigen loss with cccDNA in the liver, okay? Unless cccDNA is really transcriptionally silent, that seems to be a contradiction to me. Okay? If you have a cccDNA, of course you're going to have surface antigen in the blood. It just means your detection is not sensitive enough.
Absolutely.
I think this is a major problem that many people, especially physicians, don't recognize.
Yeah,
they don't realize.
I agree.
What we are detecting as, for example, surface antigen, the detection limit now probably is around 1 million or over 1 million particles per ml that you cannot detect. Okay? So they are not lost. It just means you cannot detect. I think there's a lot of confusion about that. It needs to be clarified. Okay?
Yeah, absolutely. I think that the choice of the markers to look at has to be really targeted to the type of intervention that we are carrying on. So as you were saying, I mean, we have to differentiate the target engagement from the readout that we want to have. Absolutely. And more sensitive assay, absolutely. Standardized assays also.
The only point I would add about this, though, is the Quant-S from Roche or Abbott are well-validated markers for functional cure. We should not use the term S-loss. We should use the term, we can't detect the subviral particles, that they're there, but they're there. But the point is it's a validated marker that has a clinical relevance for outcome. And so is it perfect? No. Is it good enough from a clinical perspective? Seems like it is.
We could go on discussing for another hour or two, but I'm afraid we do need to wrap up this symposium. So on behalf of Antonio and myself, I'd just like to thank ICE-HBV for all the help organizing and all the sponsors who contributed to this symposium, and see you all next year in Japan. So.